Hey guys, my name is Tyler Frost. I am a fourth year at Wartburg with a major in biology and minors in both chemistry and leadership. And if you can tell by the title of the video, I am a member of Chi Beta and I am going to do an article discussion. Um, the general layout, I think I'm going to talk about each part of the article, and then I'll go over at the end of the pros and the cons of what I liked about it. So a little bit of background about this. I found this article through my research that I did as a fourth year. Me and three of my peers put together a research presentation on the effect of social isolation on anxiety in guppies or schooling fish. And this was one of the articles that I found really interesting that kind of helped format how we went about things. I have my notes on it here, and I think we're just gonna continue through it, and I'm gonna try to discuss it the best I can. So I'm going to provide a link in the description of this YouTube video to so you guys can follow along with me as I talk about this article. And the title of the article is "The Behavioral Effects of Single Housing and Environmental Enrichment in Adult Zebrafish." Um, I found that abstracts are vital to the success of an article because they kind of make or break if the individual is going to continue reading based off of the information provided there. So I think we should probably start there. Looking in the abstract, within the first couple of sentences, we see their problem and what they aim to do in this article, which is to determine whether providing an artificial plant in the holding tank as enrichment influenced anxiety-like behaviors. And then they start to work into um, how they separated the fish and more of their materials and methods, um, and then into their experimentation and testing. It looks like they used the novel tank test, light dark test, and place preference tests, which I'm pretty familiar with a few of those, as I previously mentioned. So one of the things that I found interesting was the n value. Initially, looking at this abstract, it seems that the n value is pretty large, 30 for each group, which is great because it allows for a robust set of data that doesn't really, outliers don't affect it as much. So that's one thing looking at this article that um, made me want to continue reading to see how the data turned out. Continually, when looking at the abstract, we see in the last sentence, our results suggest that the addition of an artificial plant as enrichment may benefit single-housed zebrafish when social housing is not possible. And this was something that we looked at as a research group, and that was important to us because our research was to focus on social isolation, and we didn't want to have any confounding variables with anything else that would contribute to their anxiety-like behavior. So we wanted to know if enrichment would be a factor in this, and it looks like this study might help us determine that. So if we move on to the intro, we see some of their citations, and I think that they did a good job of um, laying the groundwork for why they chose zebrafish um, and why they chose to study enrichment. One of the parts that I'll pick out here is the natural habitats of zebrafish include aquatic vegetation. Providing an artificial plant has been suggested as enrichment, although it has not been evaluated objectively. And so we know that it's something that people do. We just don't know if it's actually useful or not. And so I thought that was an interesting thing to put in there. Um, more things that stuck out to me that I learned or knew already is that there are shoaling species and isolation may increase stress, which is why we see that there are control groups of grouped fish with and without the enrichment, which I thought was very important to have. Continuing in the intro, we then see them talk about the different types of behavioral tests that are often used and the ones that they used. The first that they talk about is novel tank test, which reading it, we can see that it utilizes the zebrafish's natural geotaxic instinct to dive to the bottom of the tank and then explore its environment. It's believed that the exploratory nature of the fish can be seen through its anxiety levels. And so, if they dive to the bottom of the tank and they stay at the bottom of the tank, they're more anxious and therefore less likely to explore the tank. Um, and that can be measured through the novel tank test, how much time they spend on the bottom of the tank versus the top of the tank. The next is the Scototaxis test, which is the light dark test, and that assesses anxiety-like behaviors by the amount of time spent in the light or dark area, um, kind of imitating natural shade and protection. And then last is the place preference test. And I think one of the pros about this article, and one of the things I really liked, is the use of multiple tests. It provides more backing for their findings and also allows us to look at different aspects of their test to then learn how to better implement if you're looking to research this or learning about these tests specifically. I think that they did a good job at applying multiple behavioral tests rather than just one. So if we move down to the bottom, we see their initial hypotheses, and I'll just read those. We hypothesized that providing an artificial plant would reduce anxiety-like behavior in adult zebrafish, and this effect would have a greater effect on single-housed than group-housed fish because it would serve as a hiding place and therefore provide a measure of security in an otherwise barren tank. And so that's it about the intro. I think that they did a good job laying out the steps of why they're doing this, and then ending it with the hypothesis. Let's move into the materials and methods. I'm looking for any cons. I don't have any particular things that I could pick out of the intro, but we'll keep moving forward. So if we are now moving into the materials and methods, we'll talk about this first little section called animals and housing. So this talks about how they obtained their animals for research, and it looks like they actually bred for their experimental group, and they obtained individuals under the Ayukuk policies from the Rockefellers University, and then bred them. And then it continues into the control of the system, which I think is very important, and definitely a pro is to mention all of the control variables for replication. If someone wanted to try to replicate this study, then they have all of the different things that such as the system water was maintained at 28 degrees Celsius, the pH was kept between 7.3 and 7.7, alkalinity, dissolved oxygen, all those things are super important for replication. So that's definitely a pro. Moving down further into there, it seems like they were chosen for experimentation, randomly selected at 5 months of age. That would be a question that I had, would be why 5 months? Why not 4? Why not 6? If that's just something that's that was based off of the time of the study, if that was most conducive to the research, or if they specifically chose 5 months for a reason, that's just interesting. Query that I have. Okay, so next is the experimental housing, and it looks like we finally start to talk about how long they isolated for. So it looks like they did housing environment experimentation for 3 weeks. And then again, we see for each experimental group, they have an N value of 30, which is awesome. And then they talk about the tank sizes and the overall setup of the housing, which is a really important step to make sure that the isolated fish don't come in contact. So we then go into the behavioral testing, and it goes in depth into each test a little bit more, and to what extent they went to make sure that the data was collected. We can see that they were videotaped and then watched back by a reviewer. Is interesting. Looking at this, another thing that I liked is that it says that they were blinded. The reviewer was blinded from the videos, which is a very important step to make sure that there's no confirmation bias or bias of any kind within the study. So looking at the 3 tests that they went over, they talk about them here and they provide graphics for each, which I think are really useful in trying to understand how they were laid out. Again, I have a link in the description. But the novel tank test was a 9.5 liter tank that was divided horizontally in half, and there was a marker put on the outside on that to measure the parameters. And then they randomly selected 10 from each experimental housing condition to be tested at a time. And so now we'll move into the light dark test. Again, 10 were selected from each experimental house condition and then tested. And it looks like it was the same type of tank. This one's a 5.7 liter tank that was divided vertically. And you can see the graphic provides a little more insight into it than what I can explain through words. But they were allowed to swim freely back and forth through it, and it just relied on their preference of where they sought out to be for their time that was being tested. And it also looks like the times were different, which is an interesting part. I wonder if that's just due to the nature of the test, or if that was something that was chosen by the researchers. Novel tank test was looked at for 6 minutes, whereas the light dark test was 15 minutes. And then if we move into the place preference test, like one thing that they kept consistent was the number they selected, selecting housing again. Same tank size as the light dark test was chosen, and we can see from the graphic that one side had a 17 centimeter artificial plant identical to the enrichment in the experimental housing, and on the opposite side of that was a plastic bag, an aerated plastic bag of test fish that were used, and in the center had nothing. That was a transition. And then over a 15-minute period, again, they were tested to see what their movements were, which compartment they stayed in longer. I appreciated the level of detail of the figures, and then also the figure legend help describe the diagrams, which I think is very important. Figure legends are super helpful. So moving forward to the statistical analysis, it shows that when they analyzed the data, it wasn't normally distributed, which means they had to use non-parametric tests for the results. It looks like they used the Kruskal-Wallis test, followed by a Bonferroni test to determine significance. And because they're non-parametric, we're able to see their p-value and look at their statistical significance. So moving into the results, first we'll start with the novel tank test. Oftentimes your eye kind of goes towards the figure first, and that happened with me, and it looks like the 2 bar graphs that are shown, figure A, there's a asterisk and a comparison between the group housed enriched and the single house barren, which might be something interesting. So then that kind of led me back up into the description of it, and it said that single housed barren fish had significantly fewer transitions to the upper half of the tank than group housed enriched fish. So there it is, 14 compared with 35, and the p-value under 0.05, so it was significant, which is good. But if we move forward, it looks like the rest of the studies within the novel tank test came back with no significant differences. So if we're looking at number of transitions, there was a difference between the group housed enriched and the single housed barren, which were the extremes, and but there was no difference in the amount of time spent in the upper half. Moving forward to the light dark test, again looking right at figure 5, we see an asterisk comparing the number of entries with the group housed barren, group housed enriched, and single housed barren. Excuse me. The single housed barren fish had significantly more entries to the white compartment than did the group housed barren fish. 100 compared to 57 with p-value less than 0.05, showing significance. But if we keep reading, we saw that there was no difference between the amount of time and the latency to enter the white compartment, which is interesting. I wonder if, well, this is interesting. The 6-minute trial, in the results, it says, in the figure legend, it says light dark test 6-minute trial. But if we remember back up to the materials and methods, it said 15 minutes, scored over 15 minutes. So I wonder if that's a typo or which one's correct. So I would say that the confusion of this would probably be a con, if I had to say so. But it does look like there was a little more significance. There was more significance in entries again, light dark test. Moving forward to the place preference, the final test, I see a lot of asterisks actually. So this might be the most compelling. If we read it, we see that group house barren, group house enriched, and single house barren had significantly more entries into the compartment with the artificial plant. And we can see that in the bar graphs below in figure 6a. And they prove to be less than 0.05 for their p-values. And then it looks like in figure B, when it looks at time, the single house barren fish spent significantly less time in the plant compartment than did single house enriched fish, which is interesting. And it looks like across the board, however, for the conspecifics, it was pretty average, sitting at about 150 seconds, relatively, for all of them. So that's interesting. This one does say 50 minutes. So this one is. This one does follow methods. And that's all of the data and results for this. So I think we're going to move into the discussion. They start by reevaluating what they did and restating what they did, talking about the different behavioral tests, and then they go into each of the tests, starting with the novel tank test, as to discussing their results and significance, as well as kind of backing up that the significance kind of meant something. And so if we look at the novel tank test, they go back and talk about the prolonged latency to enter the upper half of the tank and the reduced time spent in the upper half of the tank, associated with greater anxiety-like behaviors. And then they talk about a previous study that compared single housing fish with group housing fish, and the evidence supported that the more time spent on the bottom of the tank proved to show anxiety-like behaviors. Just kind of backing up what they found with previous studies. And then they kind of continue into how, well, I'll read this. The author suggested that fish housed singly or in groups may differ in development coping methods depending on their housing condition. And so this kind of talks about more into why they chose the isolation technique that they did and how it factored into their results. Continually They talk more about the statistics. So they talk about the total time spent in the upper half of the tank and how it didn't differ among experimental groups. So acknowledging that there were parts of their results that did not differ significantly is always important. Um, and then finishing it off with the results that the single barren environment influenced the exploratory as we can see. And then they back it up with the results from the bar graph saying that there were fewer exploratory transitions to the upper half of the and then we go into the light dark test. And they start by saying that adult zebrafish are scototaxic, and so that they expected this. They kind of expected their findings. And light dark test revealed altered behavior in single house barren fish, where single house barren fish entered the white compartment almost twice as often as the grouped fish. Talking again about their significant results. And one of the pros of this article, I think, in the discussion, it's sometimes it's easy for the discussion to drag on, and I think that they did a good job of being succinct in their discussion about each part. After that, they moved into the light dark test and talked about its significance and what it means. And then they finished it off with a conclusion statement, and I'll read that because I think it's actually important. Our research focused on behavioral testing and did not attempt to correlate behavioral measurements with psychologic data, evaluate sex-associated differences, related behavior responses to strain-specific differences, or measure an increased aggression. So it's kind of narrowing the focus and reminding us what was or what their research focused on. They then go into what future work could be done to improve upon this research or build upon it, which is always an important reflection piece to have as a researcher and can help others work forward in that field. They talk about how sex-associated differences may influence behavioral results, and they tried to replicate it with common groupings, so I don't think that they really singled out sexes. They kind of kept with both, but they talk about how it could be interesting to look at one sex and how it compares to the other. They continue by saying, in addition, zebrafish have been reported to display aggression towards each other. And so looking at intra-group aggression compared to between groups could be another interesting part based on enrichment and anxiety-related behaviors. And then they finish off the conclusion. And I'll read that. In conclusion, our study demonstrates that single-housed adult zebrafish in barren environments alters exploratory behavior. Single-housed fish appear to benefit from the addition of the artificial plant as an enrichment device. We conclude that using artificial plants as enrichment devices may promote the well-being of zebrafish that are singly housed. According to our findings, adding an additional artificial plant does not enhance well-being when zebrafish are group housed. And so I think from this discussion, we learn a lot about what they got out of their results and then also how it can be applied. Again, if you're looking at this from a view of someone who is looking to do research in this field or on this topic, this is a really good article because it kind of helps demonstrate how some of the more popular tests are done, but then also the value of their test in working forward, like what me and my peers did for our research, and how enrichment is important and why it should be involved in all of your isolation techniques. The only cons that I really had when looking at this article was that one discrepancy in the timing that we talked about in the results, one in the figure legend and then back in the materials and methods. They were different, which might just be a typo. And other than that, I think that it was well laid out, caught my attention well. I think it was a well put together article. And not much else to say. I hope you appreciate the discussion, and I hope you got something out of it. Again, thank you, Tri Beta, for the opportunity. And yeah, have a nice day.